briefly what is the plant breeding and what is the molecular breeding. So everybody, I think, know what is the plant breeding. It is the oldest method of crop improvement in which we just breeders select a superior plant for yield and other traits. So that will take most more than 10 years for the improvement or just creation of a new variety. Later on, just after development of molecular markers, this plant breeding approaches just take a lesser time for the improvement of the specific character, either release of new varieties. So molecular breeding has made significant achievement in crop improvement by accelerating the development of improved crop varieties with the desired traits. So it involves basically molecular biology techniques.
Sorry, sir, for the interruption, but it would be good if you will on your slideshow mode as well.
Actually, slide is just not seeing, sir. Already I have shared some slide. That is. You all are seeing my slides.
Yes, sir, now it is perfect.
Okay. So it involves the application of molecular biology techniques and tools to understand and manipulate the genetic makeup of any crop for the improvement purpose. So there is several benefits of molecular breeding techniques over the normal breeding techniques. The first one is the consistency, then efficiency, sustainability, and nutrition. These all are the important benefits over the normal plant breeding or older plant breeding methods. So, as we know, in the pre-Mendelian era, just selection method of any superior plant is the basic method for the crop improvement. And after the discovery of Mendel's
once
law,
again, sorry, sir. Sir, your voice is not clear. You have to you have to mute your mic from one device.
now I'm audible.
Yes, sir, now it is perfect.
Audio is perfect now.
Yes,
so
sir.
after discovery of Mendelian's inheritance of law, that era then started post era of the Mendelian law, and in this era, the plant breeder has just developed the hybridization method and several other improvement methods that includes mutation breeding, tissue culture, gene transfer, marker assisted selection, and thus recently just scientists has genome editing method for the specific changes in the specific trait changes in the plant system. So these are this slide shows the different pre-Mendelian, post-Mendelian, and modern breeding approaches for the improvement of the any traits in the crop plants. So this slide shows the landmark stages of plant breeding approaches. Basically, this breeding approaches has five different breeding landmarks. In the first is the human. I just discussed in the previous slide the selection and the breeding of crop with higher nutritive value involves. The second methods involve the use of human mediated cross breeding, that is the simple hybridization method and its application. And then the third method is the physical and chemical mutagenesis. And now there has specific changes with the tilling and eco-tilling methods scientists has served. And the fourth one is the genetic engineering approaches by which we can transfer the desired gene from the foreign desired gene into the specific host plant. And last one is the gene editing technology. Present time, this technology is used to for the improvement of crop plants. This slide shows the plant breeding innovation cycles, the normal plant breeding innovation cycles. And so finally, just we'll discuss the different methods or techniques for the molecular breeding. Basically, in this molecular breeding methods, nine different methods, presently more than nine methods are frequently used for the crop improvement purposes. The first one is the marker assisted selection, then the second one is the marker assisted bycross, and the third one is the marker assisted recurrent selections, then marker assisted pyramiding, and the quantitative trait locus loci mapping, then the genomic selection, then the GWAS, then the transgenic crop development, and the newest one is the genome editing. So before going to discuss the marker assisted selection, just we will just briefly discuss what is the marker. uh, uh, Without just marker, there is impossible to go for the molecular breeding. So marker is a specific DNA sequence, and basically these marker are the allelic difference or variation at the given locus in the DNA that can be observed at morphological, biochemical, and or molecular level. And molecular marker are based on naturally occurring changes on or polymorphism in the DNA sequences by which we can just differentiate two individual. And with the help of these markers, we can tag any gene or any locus, and we can also map several quantitative traits and identify also different QTLs. A perfect molecular markers has the following different characters. If we are going to molecular breeding, then a molecular marker must be polymorphic. Then if the molecular markers are codominant in nature, then that will be the very good for the transfer of any trait or tagging of any gene. Then that must be reproducible, and molecular markers then robust, then cost effective, easy to use, high throughput, and closely linked to the trait of interest. These are the following different characteristic of the perfect, which can just make a molecule to the perfect. So with the help of these molecular marker, we can go for the molecular breeding techniques. So we will just discuss the different method of the molecular breeding. Then first one is the marker assisted selection. The marker assisted selection, in the normal breeding method, we just select any superior plant on the basis of their morphology. But in molecular assisted selection or molecular breeding, we can just select any plant with the molecular markers as well as with the their physiological characters. So molecular marker assisted selection is an indirect selection of process where a trait of interest is selected not based on their trait itself, but on a molecular link to it. That means in this molecular marker methods, we just select a progeny. Either we can just uh, a plant from the population with on the basis of their morphology as well as on the molecular markers. So basically, in a molecular marker assisted selection methods, we just select two contrast parents and make a cross. Then we develop a population. Then we just do the phenotype particular traits. Then we just do genotyping with the help of molecular markers. And then we just go for the QTL mapping, linkage mapping, construction, and then QTL validation, then marker validation. Thus, this slide, these are some important steps involved in the marker assist selection. So, in this slide shows the difference between pedigree methods and marker assist selection for the early generation selections. So basically, marker research selection has just several advantages over the normal molecular breeding methods. These are the example of sweet corns by which we can just corn gene affecting carbohydrates composition of kernel has been just searched and just identified with the molecular marker assessor selection by just Hallord et al. In 1944. So these are the gene symbols and gene names, and this gene on the their different chromosome locations, and this these different genes coding proteins has the kernel phenotype affected. Just so the second one method is a marker assist backcrossing. Simply, this is a backcrossing method in which just molecular marker are used. So MABC means marker is a backcrossing methods had basically three levels, whereas a common, yeah, basic backcrossing is the only we just backcross the F1 with they are just recurrent parent and just we select the traits. But in this molecular assisted marker assisted backcrossing method, basically there are three steps. The first level is the foreground selection. In the first level, just we just uh, check the desired gene has been transferred to the concerned progeny or not with the help of molecular marker. Then again, in the backcross population, we just confirm that the population has in recombinant condition or in not. Then we just check their recombinant condition, and the third level is the background selection. In this method, the background recovery of the uh, 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 population developed has been checked by with the help of molecular markers. That one is known as the just background selection. So these are the this slide shows the background selection methods uh, of just marker back crossing methods. So just this slide shows that the P1 and P2 just cross, and then F1, then we just go for the back crossing, and just we select the recurrent parent recovery with the help of molecular marker. And the third one is the marker is a gene pyramiding. This is the technique in which multiple QTLs or gene for a single or multiple traits are transferred into a cultivar which is deficient for these traits. That means ki in by this method we can pyramid more than one gene in a single cultivars for the different characters. That may be disease resistance or agronomic improvement traits. So this method, in this method, just we cross uh, just we cross the two parents, then at the level of back cross BC3 F1 population, we just again cross, and in one population, and then again population at the same BC3 F1 just cross to just pyramid the desired gene a single progeny. So by this marker breeding approach, we can pyramid several more than not several more than one traits in a single population. So this is the different just important examples for the pyramided lines of rice, wheat, cotton, pea, barley, and soybean for the bacterial blight, blast resistance, and then leaf rust. So with the help of molecular marker, this more than uh, one gene. That means XA4, 5, 13, and 21. All these has been transferred with this uh, with the with this group, and just you say in this marker is pyramiding more than one gene has been pyramided with the help of molecular markers in a in a improve varieties that has just lacked that particular that deficient for this resistance gene. Just this slide shows the mass improved pyramided IR64 with XA5, 7, and 21. So just you see the susceptible one and the resistance one. This resistance one has a resistance for the three races for this blight. Then with the molecular marker, we just map the quantitative trait loci. That means we just that QTL is nothing that the quantitative rate locus are the locus, different locus in a genome, which can govern a specific trait like yield and disease resistance, which can govern by several gene. That means more than one gene. So just we can map how we can just map our just this one is the this slide shows the different mapping methodology. So QTL mapping just simply start with the selection of two diverse parents, just like marker assist selection. Then we can just develop mapping population that may be backcross population, either recombinant inbreed population or double haploid population. Then just we just go for phenotyping, then genotyping, then we can develop genetic map construction, and just after developing the genetic map, then we can just QTL, we can identify the QTL, and then just we validate the QTL quantitative trait locus. And this slide shows the quantitative trait locus in rice. In this, there are four different QTL for the submergence. Sub one just discovered are just located are constructed on the chromosome number 1, 2, 9, and 12. In the rice cultivars, IR72 and MADA. Then the marker assisted recurrent selection. So marker assisted recurrent selection, this is a very handful technique in which molecular markers are applied at each generation in order to target all trait of interest. It was proposed in 19. In this technique, crossing is preferred in selected individual at every crossing and selection cycle. So it involves defining a selection index of F2 or F2 derived progenies and use index to weight significant marker for target QTLs. Then recombining our self progeny of the selected individuals and repeat the procedure for a number of cycles. So this one is the normal just step involved in the marker recurrent selection in the maize. Then we go for the genomic selection. The genomic selection is a new approach for improvement of quantitative traits in large plant breeding population that uses whole genome molecular markers and combines marker data. So GS uses dense genetic markers spread across the entire genome to predict the breeding value of individual plants. By enlarging the marker associated with the important traits, breeders can make accurate prediction about performance of plant at early stage, even before they phenotypically expressed. This approach has revolutionized breeding programs, especially for complex trait controlled by multiple genes. So the important steps in the genomic selections is the development of a training population using diverse plant phenotyping, genotyping of the training population, then selection of individuals having superior GBs and on the basis of their genotypic data, progeny, then the progeny of genotypes, which are usually as a study material in the testing population, are and taken as input for the genome mix selection model and give GVS. Then the selected individuals are used as the parent for the next offspring for continuous selection and breeding. These are the just general methodology for the genomic selection. Just in the first step, training our reference population we just generate, then we go for the genotyping and phenotyping, then a statistical model we just applied and predict the trait value based on their genotypic result, and then GVS, and we can select a genomic we just select just we just select a genomic selection approach for the superior traits. So the next method is the transgenic crop improvement. That the we know the transgenic are just any crop plant that has the foreign gene in their genome, mean stable integration of any genome. The genetic engineering technique have facilitated the introduction of genes from other organism into crops, resulting in transgenic crops with improved traits. For example, the development of insect resistance by Silesthogenesis crop, just BT cotton. In India, only one just variety, just BT cotton, has been approved, commercially approved for the production of cotton. And recently, one mustard varieties from the Delhi University Group has been just approved for the commercial production. Then Supreme Court just restrict that crop for the commercial production. However, these transgenic crop have several importance over this conventional breeding approaches. Crop molecular breeding is developed crop, but and several country has been just approved transgenic crop for the cultivation, just like our just neighbor country, Bangladesh, has approved BT brinjal and other transgenic crops, and also they just uh, release golden rice for the commercial cultivation purpose. But in India, only one BT cotton has been just released for this commercial production and production for the cotton. So these are the basic method for the plant tissue is just transgenic development. We can just we can identify a specific uh, gene, then just we insert that gene into the TI plasmid, and then we can transfer to the host by with the different method, either we for with the help of TI plasmid or direct gene gun methods. Then we can select the putative transgenic, and after that we can just go for the different screening, and then uh, release for the commercial cultivation. And the recent technique is the CRISPR-Cas technique, by which we can just edit a specific trait in a this has opened up a new avenue for the targeted gene editing, allowing breeder to introduce desirable genetic variations or knock out detrimental genes in plant as well as in human. So genome editing has the potential to accelerate the development of improved crop varieties with increased precision and reduced regulatory concern compared to transgenic approach. So this CRISPR-Cas technology in India is now just is a method of transgenic, but the transgenic regulation is not just bounded this technology to release any just to release for the commercial cultivation and production. So this is the simple method for the CRISPR-Cas. Just we can design a just target and vector, then just we can introduce in a host plant for the precise changes with the help of uh, genetic engineering, means agrobacterium mediated transformation. Then we can just regenerate And after regeneration, just we can the T1 and T2 generation, we just check the changes has occurred or not. If there is a precise changes has been just found in the particular variety, then we just proceed for the environmental means checking for different traits in the environment or in just field condition. So the last one is the genome-wide just genome-wide association studies, GWAS, that is commonly known as GWAS. It the natural GWAS involves the analyzing genetic variation across the entire genome to identify associations between specific genes and trait of interest. In this method, there is no crossing or just hybridization involved. Only we just collect the wide number of germ plots, then we just screen phenotypically and just genotypically, and just we can just search our the trait of interest, and we map and we transfer that particular traits. This approach has been successful in the discovering novel genes and alleles associated with various traits in the crop. The GWise provides basically valuable insight into the genetic basis of complex trait and assist in the development of new crop varieties. This is the basic methods of the GWAS, genome-wide association studies. So in just coming slide, we just in the previous slide we just discussed the different methods for the molecular breeding. So now we will discuss the achievements in crop improvements, major crop improvements in major cereal crops developed through molecular breeding, particularly by the ICR system. What we have just developed with the help of molecular breeding approaches in the major cereal crop. Just in this slide, the two different varieties. The one is the improved Samha Mansuri, and the second one the Swarna Sub-1 varieties, which has been developed with the help of molecular breeding. And in the first improved Sama Mansuri, the trait that has been improved is the bacterial blight resistance and the gene in progress in the Pusha Basmati rice. Not Pusa Basmati, right? The Sama Mansuri, the XF5, XF13, and XF21. What marker they had used? The simple sequence repeat markers, and the recurrent variant was the Sama Mansuri, and the donor parent was the SS1113, a near isogenic line of the variety PR106. The silent feature of this improved Sama Masuri is the resistant to the bacterial blight, high yield with the excellent grain quality, trait similar to the Sama Masuri. Then a low glycemic index makes it suitable for consumption by the type 2 diabetic patient. The average gain yield of this improved Sama Masuri is the 55 quintal per hectare, and the maturity is the 130 days. The 10 days earlier than the recurrent parents. That means the Samhad Mansuri. And the second one is the one of the most mega rice variety. Swarna Sub-1 has been just developed by the International Rice Institute with the collaboration of Indian is institute. The trait improved is the submergent tolerance. The QTL intro is the Q sub-1, and the molecular again molecular markers used SSR. The recurrent parent was again Swarna sub-1. The donor parent IR 49830. That one is the FR13A. The traditional rice variety of this Odisha. From this FR31, just C sub-1 Q12 has been transferred and in progress in the Shona rice variety. The silent feature of this variety is the tolerant to the complete submergence up to 15 to 17 days. Means 15 to 70 days when the submergence has been occurred in this variety, then again it has survived, and the suitable for the planting with the aged seedling, suitable for shallow lowland and flood prone areas, and the average grain yield is the 52.5 quintal per hectare, and maturity is the 140 days. And the wheat varieties PBW 71, Unnat PBW 550. The trait improved the steep rust resistance. The gene introduced the YR15, and the recurrent parent was the BPBW115. The silent feature of is the very high level of resistance to steep resistance, and with this resistance characteristic, also they have some several other characters, good characters, like yield and maturity duration is less than this recurrent parents. And the second one is the PB W723. That is that one is the improved for that strip and leaf rust resistance. And in this variety, four genes has been introduced: YR 17, 14, 30, LR. Leaf for this strip resistance, two gene YR 17 and YR 40, and for the leaf resistance LR37 and LR57 has been introduced. And the SSSR marker for this YR17 and this 14 introgation, the SSR marker has been used, and for this in the CAPS marker. So it has again the first wheat variety developed through marker assisted selection. Means in wheat, this one is the first variety of wheat which has been developed with the help of marker assisted selection. So in maize, two for example, in this several varieties has been developed, but in this slide we just mentioned, I just mentioned only two: QPM9. One is the hybrid variety. The trait improved for the lysine and tryptophan, two essential amino acid traits have been incorporated in this maize varieties, these hybrid varieties, and this improved the quality protein, means high lysine and tryptophan as compared to the 1.2 2% lysine, mean the two times more lysine and two times more tryptophan in the popular hybrid. Average yield is again very good. Average yield is the 52 quintal per hectare, and it just matured in 88 days. And the second one is this is the Vivek QPM 9, and the trait input for the provitamin A, and it has a high provitamin A content, 1 to 2 ppm in the popular hybrids. That means more than it has just up to 9 to 10% more iron, not iron, provitamin A in this improved varieties. And then pearl millet, the HHB67, again improved trait for the downy mendo resistance. And then chickpea, Annigiri, one variety, the trait improved for the fusarium built resistance with the help of molecular markers. Then soybean and groundnut. So far, in this just ICR has developed up to 20 different varieties for more than 60 traits with the help of molecular marker. Just ICR has developed different cultivars like just rice for this bacterial blight blast, submergent rot salinity, low phosphorus, herbicide. Recently, just herbicide resistance has been developed with the background of Pusha Basmati. Uh, Dr. A.K. Singh, Ashok Kumar Singh group has been developed this herbicide tolerance varieties with the help of molecular markers. Then wheat, maize, pearl millet, chickpea, soybean. So far, 20 different varieties have been developed with the help of molecular marker or marker assisted breeding methods for different traits. These are the total, this is the combination number of cultivars improved on multiple traits by using molecular breeding. Then bacterial blight plus blast, 4 different genotypes for rice, bacterial blight plus low phosphorus 1, bacterial variety plus salinity rice in rice, one varieties. Then drought plus submergence, three varieties has been just improved, and in wheat, steep rust plus leaf rust, two variety has been released, and in maize, lysine and tryptophan, four variety has been just improved, and soybean, one variety. So total crop just species has been just seven crops improved. The highest number of crop that has the greatest number is the 43 different rice varieties has been just improved for the by the state by using molecular breeding. And then the second one is the maize, and then followed by chickpea, soybean, wheat, and pearl millet and groundnut. So so far 74 different cultivars has been just improved from 2005 to 2021 with the help of marker selection. So in the just we just I'm just going to conclude my just presentation that the molecular breeding is a game changer in a crop improvement, as this is the very faster, cheaper, and more precise method to develop and improve any crops. Some molecular breeding also bring new ethical, legal, and environmental challenges that need to be addressed just practically. The future of molecular breeding looks promising with new technology and advancements on the horizon to meet the demands of growing population sustainability, in particular in India. So thanks for the